Good morning, everyone. Today we'll continue the ongoing molecular biology lecture classes. Today we'll discuss about gene mutation and mechanism of regulation of gene expression, which is included in competency B17.3. So we'll discuss about gene mutation. So what is gene mutation? So we can discuss gene mutations under the heading of introduction. What is the history and what are the type of mutation? What is gene mutation, type of gene mutation, and molecular mechanism of gene mutation and effect of gene mutation, and what are the recent researches? So mutation, any sudden change occurring in hereditary material, is called as mutation. They may be harmful, beneficial, or neutral. So DNA is highly stable molecule that replicate within amazing accuracy. Some error of replication do occur. So coming back to the history, in the year 1901, Hugo de Vries first used the term mutation to describe the sudden heritable phenotypic change in evening primrose Oenothera lamarckicana. In the year 1904, T.H. Morgan reported white-eye Drosophila in the population of red-eye flies, and in the year 1928, H.J. Muller first used X-rays to induce mutation in fruit fly. So type of mutations. Mutations or change in the genes or chromosomes may be gene mutations or may be chromosome mutations. So under the gene mutations, there is replacement of one nucleotide by another or addition or deletion of a nucleotide. Under these chromosome mutations, may be change in the number of the chromosomes, for example, polyploidy or aneuploidy, and another one is in the chromosome mutations, there may be change in the structure of a chromosome. So gene mutation. A gene mutation is defined as an alteration in the sequence of nucleotide DNA. This change can affect a single nucleotide pair or larger gene segment of a chromosome. So type of gene mutation may be point mutation, frame shift mutation, or base substitution mutation. So what is this point mutation? It's a silent mutation, missense mutation, nonsense mutations. And under frame shift mutation, there may be insertion or there may be deletion. In case of this base substitution mutation, transition mutation and transversion mutation. So point mutation, what is point mutation? Point mutations are the most common type of gene mutation, also known as base pair substitution, change in a single nucleotide base pair. Point mutation can be categorized into three types: silent mutation, missense mutation, and nonsense mutation. So silent mutation, here you can see the change in one codon for an amino acid into another codon for that same amino acid. In case of silent mutations are also referred to as synonymous mutations. And missense mutation, here the codon for one amino acid is changed into a codon for another amino acid. Missense mutations are sometimes referred to as non-synonymous mutations. And coming to nonsense mutation, here the codon for one amino acid is changed into a translation termination codon or a stop codon. Then frame shift mutations, which is commonly asked. So what is this frame shift mutations? This type of mutation occurs when the addition or loss of DNA base changes a gene's reading frame. So a reading frame consists of three bases, each code for one amino acid. A frame shift mutation shifts the grouping of these bases and changes the code for amino acids. The resulting protein is usually non-functional. The insertion and deletion can all be frame shift mutations. So frame shift mutation, this is how original DNA code for an amino acid sequences is altered. The frame shift of one DNA base results in abnormal amino acid sequence. The insertion. An insertion changes the number of DNA base in a gene by adding a piece of DNA. As a result, the protein coded by the gene may not function properly. And what is deletion? A deletion changes the number of DNA bases by removing a piece of DNA. So small deletion may remove one or few base pair within a gene. And larger deletions can remove one entire gene or several neighboring genes The deleted DNA may alter the function of the resulting protein. So what is base substitution mutation? Here, the base substitution are mutation in which one base pair is replaced by another base pair. And base substitution can be divided into two subtypes. Number one, transition mutation, and number two, transversion mutation. So in transition mutation, a transition is the replacement of a base by the other base of the same chemical. So purine, purine, pyrimidine, pyrimidine. Then transversion mutation, here a transversion is the opposite, the replacement of a base of one chemical category by a base of the other. Then molecular mechanism of mutation. So how this mutation occurs? Maybe by spontaneous mutation, by tautomerism, by depurination, or by deamination, and maybe also induced. In case of induced mutation, maybe by physical mutagens such as radiation, ionizing and non-ionizing radiation. And heat can also induce mutation, and chemical mutagens, base analog, alkylating agents, and intercalating agents can also induce mutation. So spontaneous mutation. Here in spontaneous mutation are those which arises naturally, not through the action of a mutagenic agent. They may arise through error in DNA replication or spontaneous alteration of a nucleotide within an existing DNA molecule. So spontaneous mutation is caused by tautomerism, where a base is changed by the repositioning of a hydrogen atom, altering the hydrogen bonding pattern of that bases, resulting in an incorrect base pairing replication. And also by depurination, here loss of purine base A or G to form a apurinic site. And maybe also by deaminations. In deamination, there is hydrolysis of change a normal base to a typical base containing a keto group in place of the original amino acid. So induced mutation, during its lifetime, an organism may be exposed to a variety of physical, chemical and biological agents capable of causing damage to its genetic material. So physical mutagens, very common example is radiation, the ionizing, for example X-ray, gamma ray, cosmic ray, and non-ionizing, this ultraviolet ray, and heat break the N-glycosidic bond in DNA and result form apurinic site or base-less site. Then chemical mutagen may be base analog or alkylating agents or intercalating agent. Effect of gene mutation. So what are the effects? May be beneficial, may be neutral, or may be harmful. So what are the beneficial effect of gene mutation? May produce new version of protein essential for evolution, enhance the survival and reproductive success, turn off harmful genes. And what is the effect of this gene mutation? What are the harmful effects? There will be protein malfunction, lethal to life, sometimes stops the development of the fetus or cause genetic disorder, which is very harmful. And what is neutral effect of this gene mutation? Not significantly harm or benefited to the body. The genetic disorder, there are various genetic disorders caused by this mutation. So mutation can lead to missing or malformed proteins that can lead to disease. Some well-known inherited genetic disorders, sickle cell anemia, which is very common, and cancer. Then all these disorders are caused by the mutation of a single gene. So these are some of the signs of the mutation even in the vegetables.